Hi. Recently I've seen claims of 12 logs of dynamic range associated with another platform. Unlike Codetta Bio's Concerto system, which can quantify DNA, RNA, and protein targets simultaneously, this other platform can only measure protein targets. I thought it might be interesting to cut through the marketing spin and break down what that 12 logs of dynamic range actually represents using peer-reviewed data. When you examine the experimental results, the singleplex assays using qPCR readout demonstrate up to approximately 7 logs of dynamic range for select optimized targets. However, in the 200-plex format, most individual analytes exhibit approximately 3 logs of usable dynamic range. This reduction is not incidental. It is a direct consequence of using NGS as a shared readout, where sequencing depth must be conserved. In this Nature Communications publication, 12 logs refers to the biological concentration span of proteins in human plasma, not the measured dynamic range of the assay itself. This is an important distinction. Biology can span 12 orders of magnitude, but assays typically do not. To manage this, high abundance targets are intentionally signal attenuated using hot and cold antibody mixing. As the authors explicitly note, this compresses the per-target working range in order to prevent high abundance proteins from dominating sequencing reads. The paper reports an approximate 9.6 log cumulative dynamic range, but this value is distributed across different proteins, not achieved within any single analyte. Cumulative range describes panel coverage. Per-target dynamic range determines biological interpretability. By contrast, Codetta Bio's Concerto system assays are designed to deliver approximately 8 logs of dynamic range per target, maintained across protein, DNA, and RNA within the same workflow. We combine digital and qPCR readouts requiring absolutely no sequencing. Sample the data in 3 hours. This avoids sequencing saturation, eliminates intentional signal compression, and enables longitudinal biology without dilution gymnastics. So the question is not whether 12 logs exists in biology. The question is how many logs your assay can actually measure per target.